mark the beginning of good times for us. Get ready to jump into the bandwagon of technology, the very first online seminar being organized jointly by the MCCI and WITT. I'm sure you must have all seen the session-wise plan. We have brought together an array of knowledgeable speakers, and I'm pretty sure that you will enjoy each and every session. Today, unfortunately, Dr. Jyotsna is unable to join us due to some health issues, but instead we have Dr. Nitin Bansal of ICMR and Rajiv Gandhi Cancer Institute, New Delhi, to talk to us on serological measurements and analysis in COVID-19 context. All other speakers remain the same. Just a few general announcements to start with. Swarna will be muting everyone other than the speaker during each talk so as to minimize, the background noise and crosstalk. I request Swarna to kindly ensure this at this point of time. There would also be a question and answer session planned, and I would request you, the audience, to kindly jot down your questions and raise them at the appropriate time. Kindly put all your mobiles on the silent mode. And if you do get a call that you need to answer, we would very kindly request you to go out of the Zoom room, talk so as to avoid the disturbance to the speaker who's on the floor. Also, if you're facing any bandwidth constraint on your side, you may actually consider turning your video off so that you can listen to the good talk of the person on the Zoom room. You may also use the chat facility to raise the questions as and when they occur to you. I would now request our chairman, P.V. Jayashankar, the chairman of WITT, to welcome all the participants. It's over to Mr. P.V. Jayashankar.
Thank you, Jayanti. I hope all of you can hear me. Yeah. Good evening to everyone here. A very, very warm welcome to the second seminar event on a healthcare related topic organized by the Waterfalls Institute and MCCI. It's been just under 2 years since we had our maiden initiative on the subject of medical leadership and its challenges. Some of you were there would remember that. At that time, we are two of the most respected healthcare professionals of the country, gracing the dais and delivering wonderful special addresses. I can still remember a lot of those words. And it's our honor and privilege to once again welcome one of them, Dr. Sneh Bhargava, Padma Shri Dr. Sneh Bhargava, to this event as the chairperson. Thank you so much for joining again, Dr. Bhargava. Unfortunately for us and the healthcare community, the other luminary, Dr. Shanta, the doyen of cancer care in India, is no longer with us. May I request, as a token of our love for her, that we all observe 1 minute silence in our memory. If anyone is unable to stand comfortably, please do remain seated. Thank you, everyone. We will all miss you immensely, Dr. Shanta. As you had all noted, this is a general interest seminar today on a highly topical subject that is relevant to virtually everyone in the world today. We have been able to invite and assemble together an extremely knowledgeable set of speakers to share their knowledge and experience with us today. I would not waste time in talking about WIT or MCCI at this forum. I think so. With that, I welcome everyone once again and wish you all a fruitful evening of listening and learning. Back to you, Jayanti.
Thank you, sir. I now request Dr. Manivannan, the Managing Director, Kaveri Hospitals and General Committee member of MCCI, to deliver the opening address. A trained anesthesiologist, Dr. Manivannan's vision of making great healthcare at affordable cost became a reality in 1999. Under the wonderful leadership of Dr. Manivannan, Kaveri Hospital has successfully emulated the Japanese system of lean methodologies in every aspect of healthcare. His focus on implementation of the 5S practice has helped Kaveri Hospital win several awards in 5S Quality Circle initiatives. Dr. Manivannan, may I request you to kindly unmute yourself, and it's over to Dr. Manivannan now.
Good evening, ma'am. Thank you, ma'am. Good evening, everyone. In fact, it's a great pleasure to give the opening remarks in this very unique and very apt seminar for current situation, actually. So this seminar is being organized by Healthcare Division of Madras Chamber of Commerce in association with Waterfall Institute of Technology Transfer. I think this is the second such event with the Waterfall Institute of Technology Transfer. First one, as Mr. Jaishankar said, it is about the healthcare leadership. We had very elite leaders, and we had very good interaction. This seminar is coming at a very apt time, actually. Basically, as you all understand, when COVID has been a very challenging. In fact, we were making comments that we should forget 2020 existed in our life. In our hospital, for example, we have 7 hospitals in Tier 1, Tier 2, and Tier 3 town. All the 7 hospitals treated COVID. And right from day one, we were one of the first few hospitals to take COVID patients. When the whole world community was not aware or was not confident about the methodology, testing, treatment, and so many things, it was very challenging. And in spite of that, the doctors community and the nurses community came forward across the country, across the world. It's something which people don't know. So it created a lot of scare among a few medical community initially, but looking into people who were coming forward and working in the front line, everyone got motivated. And in that way, we were able to tackle this COVID across the world, and especially in India. People thought, India being short of some healthcare facilities, we will face a lot of challenges. But due to various factors, that includes our immunity, our disease burden, and other things, and also the infrastructure and very proactive central and state governments. So we were able to control the mortality and morbidity in this pandemic when compared to other developed countries also. So we should really be happy that we have done well as a country. And as a world, as a community, human community, we have proved that we are resilient. We can come out of any of these crises. And I think one good thing with this is, with this pandemic is, now every government or every politician, the main agenda is healthcare, actually. Earlier, as medical community, we always used to regret that none of the budget sessions or none of the election speeches, we don't find place. Or healthcare doesn't find place. But I'm really happy when, as a medical community, every one of us are happy that it has become a prime agenda for all the public figures, and which will be very, very beneficial for the community at the large. As I said earlier, because of lack of information, lack of experience, there was a lot of skepticism in treating the disease, and uncertainties, clarity was not there, resistance was there. So many things were there. But over a period of time, we learned, and we created excellent protocol. Globally, we created protocol. Locally, we fine-tuned it, and we implemented it. We all of us benefited. The same thing is with vaccine. And we all have to understand that when India has done extremely well in the recent decades with polio vaccination. We have eradicated polio in the recent times. In our lifetime, we have seen, probably senior people here would have seen smallpox eradication, other things actually. So when India has been the largest manufacturer of vaccine, which many of them didn't know before COVID. And we have been supplying more than 60 to 70% of the developing countries' vaccines across the world, actually. So whether vaccine production or vaccination implementation, our public health systems have been extremely good. But this kind of a pandemic we haven't seen. So most of the vaccinations happened in children. When we are confidently administering 5-6 vaccines to children before 2 years of age, suddenly people are a bit skeptical when vaccines are administered to adult. Of course, they we have to understand that if you take last 200 years of pandemics, probably the first pandemic took 150 years to diagnose, later treat, and vaccinations. Even say the last one, HIV, we are yet to find a vaccine because of virus changing its pattern. But this is something very unusual situation where the governments were interested, and people like Bill Gates were interested, invested money, and a lot of companies came forward, pumped in money, and came out with excellent vaccines. Of course, phase 3 trials were not completed in few vaccines, but most of the vaccines, phase 3 trials have been completed, found to be effective, and people are questioning suddenly it's only 70% or 80% effective. And we have to understand that most of the vaccines are not effective more than 85% or so. Most of the current day vaccines, whatever we take actually. So one bad thing is, an interesting thing is, today everyone started talking science actually, okay? And since they listen to some audience or listen to some unverified lectures or talks in YouTube or WhatsApp, and they think that is science actually. I firmly believe, and I keep telling my people and everyone, that we should start believing in science. Science has delivered a lot. Today, as a human beings in the entire world, we are a far, far better lot than compared to last 20, even 2 decades back. But now so many diseases are controlled. Our lifestyles have improved significantly thanks to science actually. Here, we have to trust science, our scientists, our manufacturers, our doctors. Yeah, that's why when vaccination started, all the senior committee members, we vaccinated, and normally I don't come in YouTubes and other things, but I have to necessarily do a press conference, take a photograph of me, or talk on YouTube about why vaccination is good. So I feel that as a community, medical community, or as a association, Madras Chamber of Commerce, which is a very, very, very old association, it's our responsibility to spread to the community at large the importance of vaccination and how safe it is. And with such a elite, educated speakers, and I'm sure that the confidence among participants will be improving tremendously. They can clear all the doubts. And I think this confidence should be spread to the larger community, and that will benefit. Why it is important, once again, even if you take polio vaccines, people used to ask, no, I'm living a healthy lifestyle, why should I vaccinate my child? But we have to understand that in order to attain herd immunity, all the children have to be vaccinated. That is a simple sacrifice without any great this thing. We do it for the benefit of every children in the world actually. Caste, creed, we don't bother about it. Similarly, everyone should get vaccinated. The benefit partly for us and partly for people around us and partly for the entire global community. So that is a message which we have to communicate. I'm sure this is going to benefit everyone. Once again, thank you all for being here. I'm sure it will be benefiting each and every one of you. Thank you.
Thank you, Dr. Manivannan. Your opening remarks have certainly set the pace for the seminar. Thank you once more for that. It's our honor and privilege to invite the chairperson for this event, Padma Shri Dr. Sneh Bhargava, to deliver the keynote address. An internationally respected luminary in radiology education, she is the first lady director of All India Institute of Medical Sciences, IMS, New Delhi, and the only one in its 60-year history. Dr. Sneh Bhargava also served as the chair of the hospital board of directors. She established Medical Education and Technology Center at AIIMS, designed to enable medical students to learn to teach in medicine. She is also a professor emeritus in the Department of Radiology at AIIMS. She has received innumerable awards, Lifetime Achievement Awards, and recently the Millennium Award 2000 from the Indian Radiology and Imaging Association, apart from the Padma Shri for her distinguished contribution in the field of medicine. Dr. Sneh Bhargava, may I request you to kindly unmute yourself, and we look forward to your talk.
Thank you very much. And
To
for
Dr.
all
Sneh Bhargava.
Thank you very much. I'm happy to be able to address such a gathering. And I'll start off by saying that the last year has been tumultuous for most of us. I'd like to start by bidding farewell to 2020 and welcoming 2021. We in India have faced the challenge and hopefully now in 2021 are getting past it if we remain alert and continue to practice COVID appropriate behavior, which I will call CAB, C-A-B. Our journey has its ups and downs, and COVID-19 pandemic has brought healthcare to center stage, highlighting the chronic neglect of the healthcare system, particularly primary and preventive healthcare. It has taught us how a health crisis can become a major economic and social challenge. It has been a wake-up call for policymakers on healthcare spending, which is only 4% of the government's expenditure on health, or in other words, 1.3% of GDP, which is totally inadequate and has been unchanged for the last many years. Before I come to the vaccine, I would like to introduce the virus. What do we know of the virus? It is infectious, causing mild, moderate, and severe disease. It is transmissible, causing mild, moderate, and severe transmissions. It can mutate and produce severer strains. And lastly, there is no drug available to treat or cure the disease. Therefore, our only hope is on prevention and vaccines. The government, the profession, the industry, the people have risen to the challenge, and the early and strict lockdown, though controversial, helped us to ramp up and activate an inactive bureaucracy and healthcare system. We did not even have adequate standardized PPEs, that's the personal protective equipment, or know how to don or doff it. We did not know if we had adequate oxygen supplies, ventilators, and ICU beds to deal with the onslaught. Today, we can say now, no patient died because of lack of oxygen or ventilator support, as happened in Europe. Countries who did not enact a lockdown now are facing a second wave. So far, we are safe. The trajectory of positive cases is on the downtrend, and but we are not out of the woods. The behavior of the virus is unpredictable. Therefore, our behavior has to be COVID-appropriate behavior of protecting ourselves as individuals, as transmitters, by strictly following masking, social distancing, and hand washing, which will be the norm for 2021. Also practice the 3 C's, which are as follows: avoid close contact, avoid crowded places, and avoid closed environments. Our case fatality rate has been low, which brings me to the question of immunity. When our bodies are attacked by an invader, our defense soldiers kick in to fight the invader, and the stronger one wins. The body produces antibodies to kill the invader, and this now brings me to the vaccine story. Vaccines stimulate the body to produce antibodies against the invader. They are therefore our only defense since we have no drug. The US, UK, China, Russia, and India have produced effective vaccines in record time, peer-reviewed for emergency use, approval by their regulatory authorities. All these vaccines are safe, with effectiveness varying from 60% to 91%. India, as you know, has rolled out its vaccine drive with 2 vaccines, the Covishield and Covaxin. The former produced by the Serum Institute in Pune, manufacturing the AstraZeneca vaccine developed by Oxford University, and the latter by Bharat Biotech, developed and produced in Hyderabad. Lakhs of healthcare workers have received the first dose either of these vaccines, setting to rest the question of safety. With reference to their effectiveness, we yet do not know the duration of their effectiveness, and only follow-up will tell us. You may now ask, who is at higher risk for the vaccine? It is the elderly with comorbidities. Young adults and children are at lower risks, and since 60% of our population is in the low risk category, vaccination will protect them. Each one of us has the moral responsibility for our physical health and to practice CAB if we want win the battle and break the chain of transmission to avoid a second surge. The latest Sero survey by ICMR shows that 1 out of every 5 Indians has been exposed, and at the same time up to December 2020, as assessed by antibody studies. Vaccines kick in immunity within 2 weeks of the first dose, and the second dose is a booster. But how long the immunity lasts, we still do not know. Since the currently available vaccines are 2-dose vaccines, there is no data available whether mixing the 2 types of vaccines would work better or not. And therefore, having the same vaccine at the second shot is being recommended in India. Research in the UK on mice shows a cocktail may be better. Americans are queuing up for the jab, even in their pajamas, if called at night, for the job. While the Russians, even with the lure of free vaccines, and their vaccine is 91.6% effective, they can't tempt them to shed their resistance. Awareness and effective communication appears to be necessary. In India too, there is reluctance which needs to be aggressively addressed. Vaccines are safe for pregnant women, mothers. Transplacental transfer begins around 17 weeks of gestation, and the best time to vaccinate will be the early second trimester. Now, a word about the changing colors of the virus. 3 mutations of the virus are in the news: UK, Brazil, and South Africa. The UK virus is a super spreader, while the Brazilian and South African viruses have the knack of dodging natural antibodies and make vaccines less effective. As there is a talk of herd immunity in India, should we worry about these strains? So far, we have not had an adverse effect with them, though the South African and Brazilian
Dr. Bhargava, please unmute yourself.
Yes, unmuted.
Dr. Bhargava?
Yes.
Thank you.
Can you hear me? Yes?
Yes, we can hear you, ma'am. Please go on.
So if the pandemic has ebbed in India because of herd immunity, we should be looking out for these strains. Even if the vaccines are less effective, we know vaccines prevent serious disease and death. The Sero survey in Delhi shows that every second person has been exposed. Is this herd immunity? Experts have still to say. Make a statement on it. WHO says that 60 to 70% of population needs to have immunity to break the chain of transmission. Besides Covishield and Covaxin that are being manufactured in India, and the producers of the Russian vaccine, Sputnik 5, have a collaboration with Dr. Reddy's lab in Hyderabad, which is expected to roll out the vaccine by March. Zydus in Ahmedabad are finally processing their phase 3 trials, and the Serum Institute has a collaboration with an American company to produce Novavax, which will be in powder form. The Serum Institute is exporting to many other countries, but will first meet the home needs. A mystery that I must mention: why states like Kerala and Maharashtra with better infrastructure are having the largest number of cases can only be explained by a standing immunity. It is the people in the poorer states, because of the poorer healthcare infrastructure and sanitation, causing a standing immunity. I would like to end by saying, stay safe, stay healthy, stick to science, and at the cost of repetition, follow CAB. Thank you very much. Bas?
That was wonderful. Thank you, Dr. Bhargava, for an extremely informative and interesting talk. I'm sure we are all that much wiser more on the subject. You have taught us the 3C mantra that I'm sure that all of us will adhere to keep our social responsibility. Thank you once more, Dr. Bhargava. We are certainly honored to have you here. It is now my privilege to introduce our next speaker, Dr. Shrikant Tripathi, former Director, Indian Council for Medical Research. Dr. Tripathi is currently the Director of Medical Research at the Dr. D.Y. Patil Medical College Hospital and Research Center at Pune. Earlier, he was the Director at the ICMR, National Institute for Research in Tuberculosis. Swarna, kindly transfer your control to Dr. Tripathi for his presentation. And Dr. Tripathi, I would request you to kindly unmute yourself, and we are all very eager to listen to you now.
Thank you. Should I share my screen now?
Yes, sir. Access has been given.
I'm having difficulty in sharing my screen. Just a minute.
Dr. Tripathi, you have to open the presentation in the background first and then do a share screen.
I've opened it in the background.
And now you have started screen sharing 1 minute.
Okay.
You just have to minimize your Zoom screen.
Hold on. 1 minute. Is my screen visible?
No, not yet.
Yeah, it's visible.
Oh, it's visible? Okay, great.
Yes, doctor, we can see your screen very well.
You can see, okay. The topic that I'll be talking on requirement and standards of a vaccine for COVID-19. Briefly, this topic has briefly been covered by the previous speaker, but so I'll try to be very brief on this. The vaccines are biological preparations that stimulate a person's immune system to produce immunity to specific disease, protecting that person from that disease. And there are various forms of vaccine. And if you look at the history of vaccination over the last 4 centuries, it started off with the smallpox vaccine, and after that, several vaccines have been developed. And the last efforts have been ongoing for HIV and Ebola, and now the efforts are focused on the COVID-19 vaccine. As far as the COVID vaccine development efforts are concerned, it has been really mind-boggling as to the efforts which have been taken, because in the history of vaccine development, for no other disease, we have so many vaccines in such a short period of time. We have had preclinical studies being done for animal toxicity and immune response, dose response, virus challenge, and also phase 1 clinical trials, phase 2, phase 3 clinical trials. And what we are seeing is that phase 3, some of the efficacy studies have thrown out results for in their phase 3 clinical trials. And what we have seen is that for the Pfizer and for the Moderna vaccines, results are showing almost 95% efficacy. And for the AstraZeneca, it is varying from 60% to 90% as per the results from different sources. And as far as different types of COVID-19 vaccines are concerned, we have the viral vector vaccines, the mRNA vaccines, the whole virus inactivated vaccine, and protein subunit vaccines. The viral vector vaccines and the mRNA vaccines, these work by getting into the cells and making the cells produce the COVID-19 spike protein, which in turn triggers the immune response. The whole virus vaccine and the protein subunit vaccine delivers the protein, the spike protein of the virus, and then the immune system is stimulated to produce antibodies against the protein. Now, if you look at what are the consideration to look for in an ideal SARS-CoV-2 vaccine, it must be safe and associated with only mild transient side effects. It should confirm long-lasting protection, at least for more than a season. It should protect not only against disease, but also prevent virus transmission. And it should be preferably given either as a single dose or at 2 doses. Should be able to produce in bulk quantities in a short period of time. Storage should be it should be easy to store at 2 to 8 degrees if possible, if not at least -20. -70 becomes very difficult in most parts of the world. It should be easy to be transported, hence the packaging is of crucial importance, and it should be easy to administer as well. And as far as mRNA vaccines which are available, we have the Pfizer-BioNTech vaccine and the Moderna vaccine. Unfortunately, these two vaccines have to be stored at very low temperatures. The Pfizer vaccine at -70 degrees and the Moderna at -20 degrees. We have the viral vector vaccine, which is being these are produced by Oxford AstraZeneca, CanSino Biologics, Gamaleya, and Janssen. These can be stored at 2 to 8 degrees, so that makes it easier for developing and low-income countries. Whole virus vaccine, we have the Bharat Biotech in India making the Covaxin. We have other companies as well in other parts of the world like Sinovac, Sinopharm, Medicago. And the protein subunit vaccine, which is made by Novavax, the US company, and it's also being made by the Serum Institute of India and the Chinese Academy of Sciences. These the last three groups all can be stored at 2 to 8 degrees centigrade. Now this is a busy slide, but just to highlight the fact that some of these vaccines are used extensively in many countries of the world. Like for example, the Pfizer vaccine and the Moderna vaccines are extensively used in many countries of the world. The AstraZeneca is also being used in many parts of the world. And the others, like the Sputnik vaccine, also is being used in many parts of the world. In India, we have right now AstraZeneca and Covaxin, which are available in the program. But at the same time, the phase 3 vaccine trials and phase 2 vaccine trials for many of the constructs are ongoing in India. And if you look at it, Covishield, Covaxin, the Zydus Cadila vaccine, the Sputnik vaccine, and the Novavax vaccine. All three are in either phase 2 or phase 3 trials in India. We have several other products which are in the pipeline in preclinical stages of development and are being investigated at present. Now, for the emergency use listing, we have the COVID vaccines in our country. We have 2 already approved, AstraZeneca vaccine and Covishield and Covaxin. Emergency use, as per the WHO guidelines, should be used primarily during a public health emergency of international concern. Here, use of an unlicensed product under EUL framework is based on predetermined criteria. The guidelines are given in the WHO website and also by the CDSCO in India. The national regulatory authorities of the affected countries should be involved in the EUL procedure. EUL process has been developed to expedite the availability of unlicensed medical products needed in the public health emergency situations. And the EUL is not intended to interfere with ongoing clinical trials, which can go on simultaneously, as it's happening at present. The disease for which EUL product is intended, it should be a serious or a life-threatening condition, and which has the potential for creating an outbreak, epidemic, or pandemic. And there should be no licensed products available for that indication, or the existing products have not been successful in eradicating the disease or preventing outbreaks. These are the guidelines given by WHO as well as CDSCO. The product should be manufactured using good manufacturing practices, and the EUL applicant will have to give an undertaking to complete the development of the product. As far as EUL emergency use authorization is concerned, the US FDA has given some guidelines, and these guidelines say that the vaccine recipients or their caregivers are provided with certain vaccine-specific emergency use authorization information to make an informed decision about the vaccination. This is accomplished by providing an EUA fact sheet to the recipients and the caregivers. And there are the EUA procedure for SARS-CoV-2 vaccine. These are the guidelines given. The UA should be based on an interim analysis of the clinical endpoint from a phase 3 study. The point estimate for a placebo-controlled efficacy trial, at least 50% should be there, with a lower bound confidence interval at least more than 30%. The median follow-up should be at least for 2 months after completion of the full vaccination regimen, and sufficient number should be there among the study subjects to support low risk for vaccine-induced ERD. The adequate number should be there to characterize the reactogenicity to each defined age cohorts, and more than 1,000 vaccine recipients should be followed up for assays more than 1 month after full vaccination regimen is given. And as far as WHO pre-qualification for vaccination is concerned, WHO has clearly said that vaccines that are procured by United Nations agencies and by other agencies, including Gavi and Vaccine Alliance, they should be WHO pre-qualified. And the pre-qualification procedure is listed in the WHO website. It's available, and the website address is given on the slide. And the WHO pre-qualification process assesses the vaccine quality, safety, efficacy, and suitability for use in low and middle income countries. In India, the CDSCO has given guidelines for the vaccine development. The guidelines clearly mention that the data should be generated for documenting the chemistry, manufacturing process, and controls for the vaccine. Non-clinical data, especially the vaccine-induced toxicity and also immune response, should be presented to the drug controller. In the data from the animal models should be clearly presented, as well as the clinical data, whatever is available, has to be presented to the CDSCO. The main objectives and guidelines for the development of vaccine is to generate adequate data on vaccine-related quality, safety, immunogenicity, and efficacy. All four information should be made available by the companies before it is considered for use as in the country. All vaccines developed for COVID must follow the guidelines which are required for other vaccines as well. And the guidelines are specified in the Drugs and Cosmetic Rules of 1945, the New Drugs and Clinical Trials Rules of 2019, other applicable guidelines published by CDSCO from time to time, where the manufacturer is using recombinant DNA-derived vaccine. The requirement and guidelines prescribed by Department of Biotechnology are also to be complied with. Thank you.
Thank you, sir, for giving us a wonderful glimpse into the standards, requirements, and efficacy of the vaccine. Dr. Tripathi, let me take this opportunity to thank you once more for the wonderful presentation that you have made and for the information-packed speech that was rendered by you.
Thank you.
Dear friends, the Serum Institute has been hitting the headlines globally as the largest producer of vaccine in the entire world. It stands in the forefront in the production of Covishield vaccine in India. Dr. Prasad Kulkarni, Executive Director, is a specialist in clinical pharmacology. A fellow of the American College of Clinical Pharmacology, he has to his credit over 70 publications on international journals. He is the referee for several medical journals globally. Swarna, please transfer control to Dr. Kulkarni for his presentation. Dr. Kulkarni, may I request you to unmute yourself, and we look forward to your talk.
Thank you, Dr. Jayanti. I'll just start sharing my screen. So can you see my slides? Okay, very good. So basically, I'll be talking about a rollout of the COVID-19 vaccine and more so about Covishield, which is launched by Serum Institute, and a little bit about the second vaccine that was also authorized by the government last month. So before that, a little bit again about the virus, though it has been touched upon, just to give you some idea about how the vaccine is working. So this is the SARS-CoV-2 virus. So this is not COVID-19, but SARS-CoV-2 virus, which is a spherical single-stranded RNA. And this has several antigens, several proteins, one of which is this spike protein, which you can see on the surface, right? So spike protein is the one which is really critical in making of the vaccine. There are others like envelope membrane nucleocapsid also. They are responsible for assembly of the virus. But in terms of vaccine, we are going to look at spike protein more in detail. So this spike on the surface of the virus, this is the one which actually, once the virus is inside your body in the upper respiratory tract, it binds to the receptors called as the ACE2, as angiotensin converting enzyme 2 receptors. And this receptor binding happens with the help of this spike protein, right? Again, to show you, this spike on the surface is the one which is going to attach to the receptor in our cells. So this receptor, that's why it becomes critical in the pathogenesis or in causing the disease with this virus. This has two subunits. One is S1 and then S2. So this is the S1 and this is the S2. S1 also has at the end RBD, which is called as the receptor binding domain. So this is the area which actually goes and fuses with our receptor, right? And that's how once it fuses with the receptors, then the vaccine gains entry into the cells and then starts multiplying and then starts causing all the damage. So that's why this particular protein has become an ideal target for vaccine development, right? Because if you develop antibodies against this spike, it is going to neutralize the virus and then ultimately you will be protected. So that's the whole idea behind almost all the vaccines that are under development, right? So what are the different types of vaccines that are under development? So as I said, all the vaccines, almost all the vaccines, they are aiming to expose our body to an antigen, which will not cause the disease, but at the same time, it will produce antibodies or the immune response. So which is that antigen? Spike protein. We already saw spike protein. That all vaccines are trying to deliver spike protein of the virus somehow to our body. How? So we have some viral vaccines which are either killed viruses, inactivated viruses, or weakened, which are called as also attenuated. Then we have something called as viral vector, where a virus becomes a carrier of the spike protein. We also have DNA RNA vaccines. We know that Pfizer vaccine, Moderna vaccine, they are mRNA vaccine. Then there are some which are using actual spike protein as the vaccine or putting that protein in a virus-like particle. So these are various approaches, but the underlying theme for everybody is to deliver spike protein to your body. Coming back specifically about the viral vector vaccine, and why I'm talking about this, because Covishield is a viral vector vaccine. That's the reason I will tell you a little bit about this. So this is a replicating viral vector. By vector, you mean basically a carrier. So newly approved Ebola vaccine, a few years back, Ebola was a big problem in countries like Liberia and all. So that time Ebola vaccine was developed using the same technology. This technology was developed at the Oxford University, which was subsequently licensed to AstraZeneca, and then AstraZeneca gave a sub-license to Serum Institute. So what it happens that basically you have a virus which acts like a carrier. So in our case, in case of Covishield, it is an adenovirus, but not human adenovirus, but a chimpanzee adenovirus. So an adenovirus that can infect chimpanzees. So it can only infect chimpanzees, it cannot infect humans, right? So it's not going to cause disease in humans, right? Human adenovirus can cause a significant disease to us, but because it is a chimpanzee adenovirus, it is not going to cause any harm to us. Moreover, it is made replication deficient, so it cannot replicate in our body. So it has become further safe. Now, this virus has genetic material for the coronavirus spike gene. Okay, so this in the genome of the adenovirus, you also have the genes for the spike protein. Once this vaccine is injected in our body, then our cells they get that genetic material for spike protein and they make the spike protein. Spike protein is expressed on our cells. Our immune system recognizes that spike protein and starts making antibodies against it. Right? So this is a very simple concept where a virus acts like a carrier to spike protein in a way to spike protein and gets delivered in our body. So this is what it is, non-replicating as well as replicating. The one that we are talking about is a non-replicating or replication deficient, right? So that it does not cause any harm. So coming to the Oxford vaccine, as I said, it's a chimpanzee adenovirus which is used as a vector. It has been already proven, this particular platform has already been proven safe and immunogenic for many other vaccines like MERS. I don't know whether you have heard of MERS, but MERS was a big problem in year 2011. It is Middle East Respiratory Syndrome, which is also caused by a coronavirus, right? That also caused a lot of damage, a lot of casualties in the year 2011. Then Ebola vaccine was made, malaria vaccine, also chikungunya vaccine. So all these vaccines have been developed with this particular platform. This platform was ready and therefore it could be easily translated for the coronavirus or the COVID-19, right? And that's why it was one of the earliest vaccine that could be made available, right? The speed was so much because the technology was already in place. So this particular virus, it expresses sequence for the spike protein. As I said, it is replication deficient, right? So technology transfer from Oxford to Astra to Serum Institute. This is a liquid formulation in a vial. You can see the vial here, and it is given by intramuscular route, preferably in your deltoid muscle. 0.5 ml is the dose, and 2 doses are required to be given at least 4 weeks apart. You can give them anytime between 4 to 12 weeks, right? So in terms of clinical plan, which has been already almost most of it has been covered. So a phase 1-2 trial was conducted in UK by Oxford in 1,000 people, then phase 2-3 in UK again in around 11,000 people by Oxford, then another phase 1-2 in 2,000 people in South Africa, another phase 3 in Brazil in 12,000 people, and phase 2-3 in India in 1,600 people, right? Many of these trials are still ongoing, but we already have data available. Then there are another study that is ongoing in 30,000 people in USA and also in Japan and also in Russia. Smaller studies are ongoing. So in terms of the phase 1-2 study in UK, this was a multi-center single-blind study in 1,000 people of 18 to 55 years, of which 500 received the vaccine. There were local and systemic reactions that were seen like pain, fever, chills, muscle ache, etc. But most of them were mild and there was no big problem. There was no serious adverse event that was caused by the vaccine. And the immune response was basically excellent. So these results have been published. This was the first publication on this vaccine in the journal Lancet, which is one of the topmost journals. And it exactly showed that on day 14, you had the T cell responses and IgG antibodies were by day 28 very high, and almost everybody got seroconverted. Seroconverted means it mounted every individual showed immune response to the spike protein, right? And therefore the conclusion was that it was safe and also did good antibody responses. There was one more publication which was in young and old adults, and here also the vaccine was found to be very safe, very immunogenic, few reactions, but fortunately the reactions are much less in older adults. So above 50 years, you hardly see any reactions, right? It's more in the younger age group that you see reactions. And those who receive 2 doses of the vaccine, the IgG antibodies are very high, neutralizing antibodies are very high. Even the T cell T cell responses also very high. So even in the older population, as old as you can say in the 80s, late 80s, the vaccine is working absolutely fine. So this is then the efficacy trial data came from Brazil, South Africa, and UK. All this data was pooled, and the data was analyzed. So this was almost in 23,000, almost 24,000 people. And what basically it was found that the vaccine efficacy was 70%, right? The figures that you have heard about 62% and 90%, we should not give too much credence to that, but it's the 70% is the real figure of efficacy. This was for the symptomatic disease, which also included mild cases. But remember, the vaccine was 100% effective against the severe disease. We are scared of coronavirus because it can cause severe disease, which can make you land up in a hospital and may require ventilation and ICU admission, right? And some of them actually die. So we are scared of the disease because of these severe outcomes. So all these severe outcomes are 100% prevented by the vaccine. I think that is the biggest takeaway from these trials, right? So on the basis of, and then there have been some more publications in Nature where single dose data on T cell as well as antibodies were published, and also how the booster dose, the second dose, induce multifunctional antibody responses, right? So what does this pooled data from Brazil, UK tell us? So in terms of efficacy, we have data on 20,000 people. And in terms of safety from Brazil, UK, South Africa, we have 23,000 people. So vaccine efficacy, as I said, was 70%, right? Which was highly significant, but this was again, including all types of cases. For severe cases, it was 100% efficacy. In terms of antibody responses, there it was very high, as you can see. At baseline, the mean titers were 57, whereas after dose 1, it went to 8,000. After dose 2, it went to 29,000. So the antibody responses are fantastic, just fantastic. In terms of the Indian study, which was a phase 2/3 observable and randomized study in 1,600 people, basically everybody was an adult, and 3-to-1 randomization to Covishield and to placebo in the safety cohort, and in the immunogenicity cohort also Covishield 300 and the Oxford vaccine, which was made in UK, was used as a control vaccine. Covishield obviously was made in Serum Institute here in Pune. Here again we found similar picture. The IgG antibodies were very high. So at baseline before vaccination they were 95 and 80, which was almost negligible. After first dose, they went up to 10,000 in the vaccine arm, in this vaccine arm, and almost 7,000 in the Oxford arm. After the second dose, they went further high, 33,000 and 33,000, right? So just compare 33,000 versus 95, right? So there was a hugely significant rise in the antibody titers, and these are the antibodies that are ultimately going to protect you from the infection. In terms of seroconversion, which basically means those people who got 4-fold increase in their titers compared to their baseline. So let's say at baseline somebody had 100, and if after vaccination, whether they had at least 400, 4 times their baseline titers, that is taken as a parameter of immune response mounted by the vaccine. So in terms of that seroconversion,
Unmute.
4-fold rise in the antibody titers, you can see after first dose itself, 96% and 91% actually had that rise in the titers. And after the second dose, everybody had that titer, right? So hugely, the vaccine is fantastically immunogenic. And the results of our vaccine, which is made in India, and the one which is made in UK, they are absolutely comparable. So there is no difference between the vaccine made in UK and in India, right? So that was about Covishield, our vaccine, and then little bit about the other vaccine, Covaxin, which is developed by Bharat Biotech and also ICMR, National Institute of Virology. This is a whole virion, whole virus, whole SARS-CoV-2 virus, which is inactivated on viro cells, right? So it's a killed virus. It also has a 2-dose regimen given 28 days apart. It has demonstrated immunogenicity and protective efficacy in hamsters and in monkeys, right? Also, phase 1 study was done in 375 people, phase 2 was done in 380 people, and phase 3 is ongoing. So they have a publication also in Lancet on the phase 1 study, and basically they found that the vaccine was safe and also immunogenic. So this vaccine has also been licensed, not exactly licensed. So the word that is used is EUA, emergency use authorization or restricted use authorization. So Covishield and Covaxin also, they got this EUA on 3rd January last month. And then the government, as you are aware, rolled out the vaccination for the healthcare workers on 16th January. So just 13 days after this EUA. And so far, Serum Institute has supplied millions of doses to the government of India, also to other countries like Brazil, Myanmar, Bangladesh, UAE, Morocco, Maldives, Sri Lanka. So the list is increasing every day. And till date in India, more than 54 lakh people who are healthcare workers, including doctors, nurses, ward boys, they have received the vaccine. And the vaccine has already received multiple regulatory approvals. So far, the data shows after so many people having got the vaccine that there are no safety concerns at all. And the vaccine is basically generally causing some constitutional symptoms like headache, body ache, fever for a day or two, and that's it. So this is how the data looks like, giving us good confidence that the vaccine is highly safe. So to conclude, this vaccine, Covishield, has been tested in multiple countries. The phase 1-2 trial shows that the vaccine is safe. It mounts very good antibody responses. The phase 2-3 and the phase 3 studies show that the vaccine is highly efficacious. It prevents the infection. It prevents the severe disease completely. And the Indian study also shows that the vaccine is also safe and also immunogenic in Indian population. And the rollout so far in India and also in other countries like Brazil or in Bangladesh or in Myanmar shows that there is absolutely no safety concern. And so based on this very positive data, I think we can safely say that Covishield can be a solution to the scourge of COVID-19 pandemic. So that's it from my side. Thank you very much for your attention.
Thank you very much, sir, for sharing your rich experience in this area, Dr. Kulkarni. I'm confident that all of us have learned a lot on the aspects of vaccine, the testing and its trials. It was so refreshing to listen to you and hear the process followed by the Serum Institute, given the extensive media clutter we have been subjected to. After listening to you, things seem very clear now. Thank you once more, Dr. Kulkarni. I now invite Dr. Nitin Bansal, Consultant Infectious Diseases at ICMR and Rajiv Gandhi Cancer Institute, New Delhi, to share his thoughts on an extremely important aspect of serological analysis. With a postdoctoral fellowship in infectious diseases from the Apollo Hospital, Chennai, Dr. Bansal is currently in charge of the COVID unit of Rajiv Gandhi Cancer Institute and has over 20 publications in national and international journals. Swarna, may I request you to transfer control to Dr. Bansal for his presentation? Dr. Bansal, may I request you to kindly unmute yourself, and over to you, doctor.
Am I audible?
Yes, doctor, you are.
Is my screen visible?
We are not able to see you, but we can hear you very well.
My screen is visible to you? Can you see my presentation?
Screen is visible. Screen is visible, doctor.
Okay. Okay. So good evening, everyone. And first of all, I would like to thank everyone to give me an opportunity to present some aspects of serology in COVID. I would like to begin with a disclaimer. The disclaimer is that I am a person of applied science. I am not a person of basic science, so I am not that learned in terms of basics of virus or basics of the serology. I am basically a clinician, so I apply what report I get. So this is a picture representing the difference between basic science and applied science. And that's why my view on a topic will be very different from a person who is sitting in the laboratory and doing basically using their expertise in developing vaccines or developing serological tests for the benefit of the clinicians, basically. So this will be the framework of my small talk. What are serological tests? Dynamics of serology in COVID, best utilization of serological tests, experience of using serology test at RGCI, pitfalls of COVID serology. So what are serological tests? Serology is basically the scientific study of serum and other body fluids, and it is a diagnostic identification of antibodies in the serum. So it can be used through various methods: ELISA-based method, agglutination-based methods, precipitation, complement fixation, fluorescent antibodies, and chemiluminescence. I'll not go into too much detail into everything because this is, first of all, beyond my expertise. Secondly, it will make the topic more lengthy. So coming to basic school immunology, which we learn in our school, whenever we get an antigen exposure, in this case, we can take it as a COVID virus. Our body mounts an antibody response in response to the antigen or the foreign body. And one of the first antibodies to get mounted is IgM antibody. And then as the time passes, this IgM, there is another antibody which comes in, which is known as IgG. As you can see from the graph, as the time passes by, the IgG becomes predominant and the IgM virtually wanes off slowly. The more and more antigen exposure we get, the stronger the antibody response we will get. I'll come back to this in a little while. So we can measure these antibodies in a patient's blood or a body through various methods. One of the common method is using this CAR test or immunochromatographic test, where a spot of blood from the finger prick is used and put on this well, and then the blood basically is imbibed onto this card, and subsequently it is read by the lab technician. This is the control arm, and this is the control strip, and these are the positive tests. Then there is an ELISA-based test, which is this is the ELISA-based test. This is the precipitation we commonly use to know which blood group which we are, and this is the chemiluminescence test, where a special enzyme is used, which basically lights up on throwing away the light, and it basically binds to the antigen antibody complex. So basics of immunology. To understand serology, we need to understand what are the basics of immunology. Our body's immunity is divided into two aspects: innate response and adaptive response. Innate response is more of a primitive response, which is found in lower animals also, and it is through like natural killer cells or macrophages, whereas adaptive response is a more sophisticated response to an antigen or a foreign body, which again is of two types, by T cell and by B cells. B cells are the one which are responsible for production of antibodies and subsequent utilization of the pathogen, whereas T cells are the one which are responsible for the intracellular killing of the pathogen and forming memory cells. So T cell is a more sophisticated version of immunity as compared to B cells, but both are part of the adaptive response. So just in a little bit more detail, this is almost similar to the previous diagram which I have shown you, that whenever there is an antigen exposure, apart from the innate response, an adaptive response takes place, and we form a lot of B cells. These B cells are naive B cells, and they start forming antibodies. So and antibodies are used to neutralize the virus. Some of these B cells form into long-lived plasma cells or B memory cells, which stay close to years into the body. So whenever we get a repeat infections, these B cells become activated and neutralize that infection subsequently. So antibody levels in the blood, which will subsequently come down as the antigen exposure wanes off. But when you get a repeat exposure, antibody levels will again rise up. So what are the dynamics of serology in COVID? So this diagram has been put forward in various presentations, and I think we know the virus more than what we know our body of. So just to be very brief, it has a spike protein, it has a nucleic acid protein, and then there is a membrane, there is an envelope, and then there is a genome of the virus. Why I'm showing is that because our body forms antibodies to the specific proteins. One of the two common specific proteins which we measure are the antibodies to nucleic acid and antibodies to the spike antigen. So majority of the antibodies are produced against the most abundant protein of the virus, which is the nucleic acid protein. Therefore, it is the detection of these antibodies would be the most sensitive test. That is, it will present in most of the patients. Whereas, since as was described by the previous speaker, the antibodies to the spike protein, which has a receptor binding domain, will be more specific and will be more predictive for neutralization, because that is the portion of the virus which binds to our body cells. So the antibodies against that protein will be more specific and neutralizing. So coming to the dynamics per se, if you can see, as we get the infection on day 0, it is almost by day 7 we start getting an IgM response, and almost after 10 to 14 days of the infection we start getting an IgG response, and it is expected that IgG will remain for quite a long time. Till now we are almost like an year into the pandemic. The studies have shown that it remains well detectable for around 6 to 7 months. Subsequently also, as the studies will come, we will know whether it actually remains there or not, but as of now we can say it remains detectable in the body for more than 6 months. So there are various methods to detect the antibodies. There are various kits available worldwide. Various kits are utilized in various labs to make the diagnosis that their body has antibodies against it or not. The reason for putting up with this slide is that we need to be aware which test is being used to measure the antibody, because each test, first of all, targets different type of antibody in the body. So subsequently, sensitivity and specificity will differ. Not all the tests are comparable. So what is the best utilization of serology test? They can be utilized for diagnosis. It can be utilized for therapeutics. It can be utilized in epidemiological surveillance, and it can be utilized to measure the vaccine response, as was shown by the previous speaker, how almost all vaccinated patients were subjected to serological tests, and vaccine was found to be immunogenic. So coming to diagnostics, diagnostic tests, serological tests are not gold standard for diagnosis of COVID. It is the RT-PCR test which remains as a gold standard, but it may have a supportive role in terms of diagnosis. What happens with RT-PCR is that if the patient presents very late to you, like more than 10 days of illness, the RT-PCR may be falsely negative. In that case, a positive IgM COVID test may help and may give you a supportive diagnosis of COVID. And on those patients we are not able to perform bronchoscopy because of infection control reasons, or because the patient is very sick on ventilator, bronchoscopy is not possible, we can do an antibody test and get to a supportive diagnosis of COVID. Or we can do a retrospective diagnosis, but for that we need a paired serum. That is the serum at the time where the patient was ill, and the serum when the patient has recovered, and we can see that the patient has seroconverted, like he was antibody negative initially and now become antibody positive. So maybe he had a diagnosis, he had a COVID earlier. Therapeutics, although it is debatable how we are using antibody tests for therapy, but it has a role in therapeutics. As we all know, there is some role of convalescent plasma therapy in moderate to severe COVID, and it is generally based on the measurement of the antibody levels in the donor plasma. So only when the donor plasma has some particular amount of antibodies, good amount of neutralizing antibody, then only it is beneficial. And also the recipient, the patient, should be antibody negative. This is one of the major papers which was published very recently in NEJM, which showed that the transfusion plasma with higher antibody levels was associated with lower death than transfusion with lower antibody level. That is, the donor should have a higher antibody levels in the plasma itself. It will lead to better outcomes. And ICMR also recently gave us a sort of an algorithm how to optimally utilize convalescent plasma, in which also they mentioned that antibody test can be done from the patient. If the patient is negative, the chances are there that the patient may benefit from the convalescent plasma. So this is what we were following also. And then epidemiological surveillance, everyone keeps on reading in the news and newspapers that so and so surveillance was done. So various surveillance has been done by various countries and also by our own country. This is the photo of a surveillance which has been done conducted last week in New Delhi, where we can see most of the districts have attained a seroprevalence of around 50 to 60%, like coming close to herd immunity. So this have been done starting from August. We have been doing serosurveillance in our own country. So coming to experience of using serological test or RGCI. RGCI, basically, we are a cancer institute, and we are a dedicated cancer institute, and we basically have a lot of immunocompromised stores. A lot of our cancer therapies contains rituximab and some other anti-B cell or sometimes even anti-T cell therapy. So antibody response is poor. So as far as diagnosis of COVID is concerned, we did not rely on serological tests for diagnosis of COVID. We relied on the PCR tests. But we used serology. We used serology for the transfusion of plasma. There was a lot of shortage of plasma. There was a lot of hype of plasma. So how to optimally use the plasma, how to rationalize the use of plasma. This is the flowchart which we followed. Just briefly, I would like to describe this. As soon as the patient came with COVID, who was supposed to, cancer patient who came with COVID, who was on oxygen, step 1 was to start on standard COVID therapy, that is remdesivir, steroids, and anticoagulation. And step 2 was to assess the patient's clinical status in 24 hours. And if he's not improving in terms of oxygenation, we would check his COVID IgG levels. If COVID IgG levels were negative, most of our patients turned out to be that they were negative. We could arrange for a donor with good neutralizing antibody levels, and we could help them. We transfused nearly around 150 to 200 patients with plasma, and they could benefit from that. There is also a role of of serology in infection control. One of the major problems which comes in infection in immunocompromised sources that they are prolonged shedders of COVID virus. So it becomes very difficult for us to when to safely remove them from isolation. Although there are guidelines which say that after 20 days the virus becomes virtually non-infectious, but still it is a little dicey when to remove them since the subsequent patient also is immunocompromised. So what we followed at RGCI was, if there is a persistent positivity of COVID RT-PCR, that is the patient is improved, but still the COVID RT-PCR is coming positive. So if we would look into three factors. If the patient was asymptomatic, he had spent more than 14 days or 20 days if it is neutropenia in isolation, or his COVID antibody levels are good, that is, he's having more than 1/64 neutralizing antibody levels, we would remove him from isolation and continue with subsequent cancer care, that is, giving him further chemotherapy, going for radiation or surgeries and all subsequently like that. So what are the so all is not well with COVID serology. COVID serology, a lot of pitfalls. First of all, you need to know what kind of a kit you are using and what is the sensitivity and specificity of that kit. It will vary. Timing is of utmost importance. If you do the serology test very early in a particular patient, it will come out to be falsely negative. If you do it very late, it may just indicate past infection. And it can't replace PCR-based tests. PCR-based tests are mandatory for diagnosis of COVID. And as we move along into pandemic, and the pandemic becomes endemic, that is, most of the people living in this world get infected or exposed to COVID, the utility of serology will come down, because all of us will have antibodies someday or the other, and now subsequently vaccinations come also. So we expect that all of us will have a positive IgG or serologic-based test. So one of the major important questions which comes to with respect to serology in terms of vaccination is, should we use serology prior to vaccination? That is, I'm going to get a vaccination today. Should I get my IgG level tested? If it is negative, then only should I get vaccinated, or should I not do it? So as far as guidelines and logic says, you should not use serology-based test prior to vaccination, because B-cell immunity, as I showed, is the only one arm. There is a T-cell immune arm also. Even if you are negative, it does not mean that you are not exposed. And even if it is positive, it is bound to wane off. So you need to have a more sophisticated, that is more advanced form of immunity, which is through the T-cell. So vaccine not only gives you antibodies, it also gives you T-cell immunity. And serology tests are not 100% specific. Your serology test may come out to be positive, but it may be because of some other virus. It may be falsely positive also. And if at all you would have got an infection earlier and your antibody is truly positive, still vaccine will act as a booster. So as far as now, there is no guidelines, no recommendation on whether to said you should use your antibody test for prior to vaccination. You should just go ahead with vaccination. So to summarize, antibody levels are sometimes like a very small child. It looks like all over the place. But when you have a proper guidance, when you use it properly, it makes more sense. Thank you.
Thank you, Dr. Bansal, for your fascinating overview of the serological aspects, something that most of us were not aware of. Your input on serology prior to vaccination, I'm sure, would be very handy to every listener today. Thank you for that eye-opening part.
Thank you, ma'am.
It is now my pleasure to invite our final speaker of the day, Dr. K.S. Kardam. With a doctorate in intellectual property law, Dr. Kardam headed the patent office at New Delhi before becoming the senior joint controller of patents and designs. He's been a part of the task force constituted for traditional knowledge digital library and has represented India on many an occasion to the WIPO. Dr. Kardam has published several papers and delivered talks at national and international seminars in the area of IPR. Dr. Kardam, may I request you to kindly unmute yourself, and we look forward to your talk.
Thank you, ma'am. And first of all, good evening to everyone. And I would also like to thank Dr. Swaminathan and the Madras Chambers of Commerce and Industry for inviting me to share my views on the issue relating to vaccines, especially the intellectual property rights. So, as you know, the intellectual property rights are the rights granted by for the creativity, and as you all know that vaccines have been made and developed and invented in a very short period of time. They are very, as you must have, everyone has seen that the main companies which are involved in inventing or discovering these vaccines are Pfizer, BioNTech, and our own, Oxford University, and with the help of AstraZeneca, Moderna, and the Russian companies and Chinese all have developed vaccines. And these vaccines, in fact, the IPR issues are very, very important, because normally this IPR, intellectual property, especially the patents, are granted to the individual entity, maybe a company or a inventor, whosoever apply for patent. But as you all know, maybe I would like to say that when I did my some survey, then I found that BioNTech has applied for about 13 patents for various kind of inventions like lipid plus mRNA vaccine, and there are 9 vaccines based for the pharmaceutical compositions, and 1 for mRNA kind of vaccine, and there are 3 more having the mixed admixtures or the compositions. Similarly, the Moderna has obtained around 7 patents relating to vaccines, and very recently they have got 2 patents recently in January 2021. So there are patents obtained by them, but we look at their financing issues, like this Pfizer and BioNTech. They have got a lot of funding actually from various sources, especially BioNTech has received around 375 million euros from German government, and another 100 million loan from the European Investment Bank. And moreover, this particular company has used the spike protein technology developed by scientists from US government National Institute of Health. So the now issue is who is because so much of money received from the government, then who will be owning these patents? As on today, I have seen these patents are applied in their name, BioNTech, but there is no name of the US government or German government in these patents. So, but in US there is a Bayh-Dole Act by which the government can exercise their powers, to share the revenue and other kind of things. Similarly, this Oxford University vaccine has also got a lot of funds from the various agencies, like UK government itself has given around 65 million euros, and US, there is an organization which has also given 892 million euros, and there is an organization SEPI also given some money to them. So overall, they have got around 963 million euros for development of this vaccine. So again, since Oxford, I am not aware whether they have applied for any patent, because they have nowadays a publication of any application, it takes place about after expiry of 18 months. So I try to find it out, but still I am not able to get there what are the status of their patents in India and also abroad. As I said, Madonna also got the 7 patents. One of them was recently granted January 2021. But all after having developed all these patents with the help of the various governments, contribution. So we really have to understand what will be their role and all. But by these donations or maybe as a loan, the situation I think is going to become very worse, particularly in the sense of stockpiling of these vaccines. You must have heard about the stockpiling of these vaccines by high income countries through pre-purchase orders. They have made some agreement with these companies. Like 82% of the Pfizer 1.35 billion vaccine stock to be made available to the by the end of 2021. They have been pre-booked by various countries. Like United Kingdom has booked 40 millions. US has booked 100 to plus 500 millions doses. EU has booked 200 millions doses. Japan and Canada, which have so they have also booked. So it is only they are representing only 14% of the total population of the world, but they have already booked 82% of the vaccines. So wherever the developing country will go to have vaccination in their country. So similarly, the Moderna vaccine also about 78% vaccine has been booked by these countries. AstraZeneca too has committed 100 million doses to UK. So the now issue is whether by such patenting or IPR, activities, whether the developing countries would be able to get a vaccine at low price, low and affordable prices. That too within this year. This is a very, very important issue. And to address this issue, in fact, India has made a proposal in WTO Council to have a waiver of implementation of certain provisions of TRIPS agreement by denying the IPR to certain years of time. But this proposal by India and South Africa has been opposed by many countries, especially the developed countries, on various flimsy ground that it will be a detrimental to the innovation activity being done by these companies. Especially, and we are very surprised that the country like Brazil has also joined this kind of, with the developed countries, to oppose this kind of proposal from India and South Africa. But, these proposals are now under study, and the last meeting was held in January 2021, but it concluded without any further result. So, and there is another effort done by WHO that is known as COVAX, that is a global collaboration for access to technology, access to the COVID technology access pool. So, but again, here also they are not able to, implement this successfully. In fact, under this program, the WHO, targeting to provide or to distribute the vaccine across 172 members countries. And this particular COVAX program being made by raising the funds from UK, EU, Italy, and even certain Bill Gates Foundation, etc. But I don't know how far these efforts will be successful, and if they are successful, then only the developing countries would be able to get these vaccines. As far as India is concerned, when there are, Indian Patent Act provide a lot of provisions to ensure the access to these medicines. There are various inbuilt provisions in Indian Patent Act. One of them is Section 47, which provides that the government can use any patented inventions or the relating to drug for the distribution to the hospitals and dispensaries. I mean, maybe they can consider later on if the situation arises. There are also provisions relating to compulsory license. If the drugs are, the patented drug is not manufactured in India or not available at reasonable price or reasonably affordable price, or it is not meeting the requirement of the public and not commercially worked in India. So there are three grounds for general compulsory license to be granted. And there are two more very interesting provisions under the Indian Patent Act. One is relating to special provision for compulsory license by the central government in case of circumstances of national emergency, or in case of extreme urgency, or in case of public non-commercial use. And there is another provision in the Act which provides that in case of any pandemic situation or maybe public health crisis, the controller can grant a compulsory license without even providing an opportunity to the right holder. So if this kind of situation comes, the Indian patent law is quite sufficient to deal with such situation. If the prices are high, if there is a scarcity of the medicine available to the Indian people, or so, although there is previous speaker have explained how the India vaccination drive is going on, and for developing countries also there is a Doha declaration provision which enables any country to provide to export the patented pharmaceutical product to the countries which have which do not have, I think, manufacturing capacity, or even if they have very insignificant or insufficient facility for manufacturing the pharmaceutical product. So if the situation arises, even if the efforts by WHO do not work in the favor of the developing countries, but India, I think, since India has a lot of experience in manufacturing the vaccines, and our government already started taking initiative to provide certain dose of vaccines to various countries. I think this is the one provision under which that is Section 92A, where the government can exercise this power by manufacturing the patented drugs in India and export to those countries wherever the facility of manufacturing is not available. So I think if the efforts on WTO level, like waiver issue, which do not work in India's favor, because of the questions, or because of the objections raised by the developed countries. So India, when it comes to the developing country, I think India should exercise this provision and make available these vaccines to the developing countries. And another proposal is also pending at WTO level, that the transition period, as you all know, that the TRIPS agreement provides various transition provision to implement the TRIPS agreement, and the least developing countries are provided 10 years time to implement the TRIPS provision. They have been extended last time, and again they are requesting to extend up to 2033. So these are the various, I think, provisions. Although the vaccines are now patentable in India, very much patentable, although I think before 2002 they were not patentable, but now they are very much patentable. There are a lot of vaccines applications are received, and a lot of patents have also been granted. But of course, the method of treatment of human beings or animals are not patentable. While granting the patent, these provisions are considered by the patent examiner. And besides patent, there are other intellectual property rights like by the name by which these vaccines are to be sold in the market. They are covered under trademark. And already I think there is a court case going on against Serum Institute by some Pune-based company where they registered a trademark Covishield prior to the Serum Institute use this trademark. So such issues are also are now being challenged and being faced by companies by their trademark. So and there is another provision under the Patent Act, like the power of the central government to use invention for the purpose of the government. So there is under section 100, which also empowers the central government to use the inventions for the government purpose. So I think India, not only India is quite strong in implementing those provisions. As you all know, the India granted first compulsory license against Bayer patents, and if time comes, I think these provisions can be used at any time when the situation arises and demand. And with this, I would also like to say that the even developing countries, as I said previously, if the talks are not successful, India is ready. India will be, I think, ready to exercise the special provisions for exporting of pharmaceutical patents, pharmaceutical patented product to developing countries wherever there is a scarcity of manufacturing or lack of manufacturing. So with these, I conclude my short talk. Thank you very much.
Thank you, Dr. Kardam, for sharing that very informative perspective on the IPR related aspects, which are very important to any developing economy and particularly to the entire world. As one of the observers rightly pointed out, the Indian patent law is certainly very encouraging to note, and we have really been enriched by your talk. Thank you once more, Dr. Kardam. Now we move over to one very interesting aspect as a culmination to all the talks that we have been listening to now. A panel discussion followed, which will be moderated by Dr. K.V. Swaminathan. Dr. Swaminathan is a retired technocrat who founded the WITT institution at New Delhi in the 1990s. Healthcare has been his particular focus, and he is a part of the governing council at both Sitaram Bharatiya Hospital and Rajiv Gandhi Cancer Institute and Research Center, both at New Delhi. Prior to that, he was the advisor to the Ministry of Science and Technology and also the Controller General of Patents and Trademarks and the director of the Cement Research Institute. It's over to Dr. Swaminathan. Doctor, please unmute yourself. Thank you, Doctor.
Can I start, Jayanti?
Yes, sir, you may. We're all with you.
Okay. First of all, I enjoyed listening to the various speakers, and each one did their job exceedingly well within the times that we had prepared for them, and also they have stressed entirely different points to show the expertise in this field is not in one field but in very many dimensions. So at this stage, the moderator's job in a panel discussion could be just to ask any one of these speakers, would they like to add a point or two on the basis of what they heard from the other speakers. So may I start with Dr. Manivannan? Do you think you have any question to pose to any of the other panelists?
she has to tell that lady.
Then please, the floor is yours.
You have to unmute yourself.
You know. Now, otherwise, can
Okay,
I
give me-
then ask Dr. Sneh Bhargava, would you like to ask for a clarification or would like to supplement something on the basis of what you heard? Dr. Bhargava? Jayanti, am I being able to communicate to them or?
Anand. That is Anand.
Okay. Jayanti, I saw Bhargava. Dr. Bhargava, would you like to say something?
Mute yourself.
I have to mute myself here or?
No, Dr. Bhargava.
Correct. Okay.
Okay, we could move over to the next speaker, Dr. Swaminathan.
Okay. Dr. Tripathi, would you like to supplement something or ask a clarification?
Yes, sir. I just have one. There's one issue which a lot of people have been asking, and that is if someone has already developed COVID, what should be the guidance given to them? And, when should they take the vaccine? And is one dose enough or 2 doses have to be given? There are some reports which say that if somebody has developed COVID, that CDC guidelines says if 3 months have passed, you can give the vaccination. But so is it a single dose to be given, 2 doses to be given? All these issues are troubling a lot of people who have had COVID infection in the past. So this topic definitely needs further clarification. Maybe more research is also needed on this topic.
Now, from what I heard from Dr. Bhargava a little earlier, one comment she made was, sometimes a cocktail will do better. Well, she didn't have time to explain it. Maybe that may probably one segment of the answer.
I want to
question.
speak now. Huh?
Speak. Yes, Dr. Bhargava, you're on the floor now.
Yes. Cocktail means you don't have the same vaccine as a second dose. And
Right.
that will give you more immunity than giving the same shot. This is experimental still, and no human trials have been done.
Oh, I see. No human trials. Good. Dr. Tripathi, the one segment of the question has been answered. The remaining part of it is what period you should. I think AstraZeneca, if I remember right, had mentioned that if they give it after the 4 weeks trial or something of that, the 4 weeks intermediate gap, if the second one is given, probably they had something like an effectiveness in the range of maybe 65, 70 of that order of magnitude. But if that gap is increased, probably they were coming closer to 90, 95% efficiency. So there seems to be some logic in not necessarily hurrying to take a second dose, but seems to be an advantage to delay it, to take it to a little later stage. I don't know how far the experimental data or the data which may be in phase 4 trial sometime, that when we get this data collected together, we may be able to give an opinion. Would anybody like to give some answers to that? Maybe Dr. Tripathi, no, Dr. Kulkarni may be able to do, because they have some problem with varying efficacy starting from about 70 to 95 in their experience. Dr. Kulkarni?
Yeah, right, sir. I would like to comment to Dr. Tripathi's question first. That as we are aware that yes, those who have been infected in the past, it is recommended that they should get a vaccine, because we know that reinfection can happen. and for some it might be severe also. So vaccine is recommended, whether 1 or 2 doses. At the moment, 2 doses schedule is recommended. I think he's talking about the recent report where 1 dose has also been found to be very effective. So that data is there, but as of now, there is no such recommendation. So we'll have to probably wait for some policy recommendation from the various world bodies. At the moment, 2 doses, and of course, at least 4 weeks apart, that that schedule should be given. And what we are recommending is when to give after the infection. So at least 2 weeks after the clinical recovery from the infection. So there should be a gap of at least 2 weeks after the recovery when you can give the vaccine, the first dose of the vaccine. Then as regards to the interval between 2 doses of the vaccine, as you rightly said, sir, the more the interval, the better is going to be the immune response, and that holds true for all vaccines, not just for COVID vaccine, but for any vaccine, you will always see a better response. Now, the only downside of that is, what if you are not protected with the first dose, and then before even getting the second dose, you get infected? What happens, right? So it's you have to have a balance between having a better immune response and then also having the response earlier, as early as possible, so that you are protected, as soon as possible. So that you have to keep a balance between the two. And the response, the efficacy figures that we saw for Oxford vaccine, the difference 62% versus 90% that everybody has talked about, that whether the difference was because the half dose was given as the first dose and that gave better protection. So it is clear that it had nothing to do with half dose or full dose. It was basically because in the half dose group, the duration between 2 doses was longer. Most of the people in that particular group had received second dose almost 3 months after the first dose. And that was the reason why you got better protection in that particular population. So yes, more the duration between 2 doses, it is going to be better. But again, you have to weigh the risk that you might get infected before getting the second dose.
Thank you. Then Dr. Kulkarni, it is your chance now to say whether you would like to supplement or to ask for a clarification from the others on what they said.
No, sir. I think all the presentations were very clear and really gave a very good insight on their topic.
Dr. Bansal, are you available?
Yes, sir, I am available, sir.
Well,
I would like to ask a question, sir.
I especially thank you because you were not given much time to prepare for this, but I think you made a good presentation and you were a little apologetic to say that from the theoretical side, you may be not very up to date, but I thought you did a very good job. All your diagrams and other graphs which you presented shows that you are well entrenched into the subject.
Thank you, sir.
Anyone want to answer something?
Sir, I would like to ask a question to Dr. Prasad, sir.
Oh, welcome, welcome.
So I would like to ask whether
this portion.
Yeah,
Yes,
my question is whether we can give this adenovirus vaccine to the immunocompromised host. Since it has an adenovirus, there is a concern that it might be replicative. Although I know that it's a non-replicating virus, are we sure that it can be safely given to the immunocompromised host, particularly stem cell transplant hosts and post-chemotherapy hosts?
Right. So as you rightly said, this is a replication deficient virus. Also, this is a chimpanzee adenovirus. So the host for this virus is chimpanzee and not humans. So that makes it still more safer for humans. At the same time, we do not have enough data from clinical trials on immunocompromised people. So we'll have to be cautious. The immunocompromised people, like what you're talking about, like stem cell recipients or organ transplant people, they are in fact at a high risk of severe COVID-19 disease, as we all are aware, right? So the physician has to weigh the benefit risk analysis in its judgment, that if the benefit is the protection from a very severe disease which can be life-threatening, then I think it is worth giving. right? So that decision has to be taken by the treating physician. At the same time, we should also be aware that that the immune response to the vaccine may be a little lower as compared to the immunocompetent people, naturally because of their immunosuppressed condition. But still, it will give definitely some degree of protection. So the treating physician should take a decision, considering that the benefits are very, very high.
Thank you, sir.
Dr. Kulkarni,
Yeah.
Dr. I have a clarification to seek from you. When we talk of 2 doses, when we talk of 2 doses, they are 50-50, or it could be varying between 60 to 40 or 25 to 75, something like that, 2 portions. In which case, which will be the bigger dose, first one or the second one?
No. So the currently licensed regimen includes 2 full doses, which is 0.5 ml volume that contains 5 into 10^10 of the virus. So that's a full dose for both the doses.
Okay. Now, Dr. Kardam, you have a chance to seek any clarification from any of the panelists.
Thank you, Dr. Swaminathan. Actually, I saw the presentation of Dr. Kulkarni, where he mentioned that there are certain clinical trials of some drugs are still going on. In one of the case in phase 3, there are only 365 people registered for phase 3. So whether with this kind of number, would you be clarify whether this will be safe? I mean, on these number, whether the vaccine can be brought to the market for vaccination, and also the phase 3 trial, I mean, or the various companies are not yet over. The current vaccination is safe, because a lot of people have, doubt about this efficacy and the safety of the vaccines.
Right. So I think as far as Covishield is concerned, I think I showed you a lot of data on almost 25,000 people, and which has clearly shown the safety as well as efficacy in different countries. So there is ample data on that vaccine. As regards the other vaccine, their phase 2 trial was on 300 something population, as you rightly pointed out. They also had generated safety data on I think more than 20,000 people in their phase 3 trial. So safety data was also available. So that all this data they took to the regulators, and based on the pandemic situation, they were given the emergency use authorization. So I think the regulators must have taken all factors into consideration. So we should not worry about the safety part of it.
Thank you, Doctor.
Well, Jayanti, but now the floor is back to you, and you may seek a question from any other people who have been listening to us outside the panelists.
Thank you, Dr. Swaminathan. I cannot fail to mention here that this moderator, Dr. Swaminathan, did so much better than the India Today or Republic TV, where we have all the speakers, clamoring and cluttering to talk. And it was a very informative panel discussion, and all of us got to benefit through this. And thank you, Dr. Swaminathan, for this. We now open the floor to questions.
Can I
request you to kindly use the raise your hand option using the Zoom screen, or perhaps you could just unmute yourself to ask the question after the organizer, after we actually identify your name for the question. I see a lot of questions on the chat.
Jaishankar here, can I ask the first question?
Yes, the chairman of WIT is ready to shoot his question. So let's have him on the floor first.
Yeah, one area of things which I've been reading about is the testing of the vaccines for specific genetics. The Chinese mongoloid genetics, the Indian genetics, the African genetics, the white genetics. I mean, except for the Oxford thing, I don't see much of Indian genetics testing. Is that an area of concern for us? Dr. Kulkarni, Dr. Tripathi, anyone?
So I think for Oxford, I think we have data from all the races and all the ethnicities, right? So I think there is no concern about that. Other vaccines, I think, are still generating that kind of data. But usually for vaccines, the racial and ethnic factors do not really make so much difference. That has not been seen. They may make a difference for some drugs, not all drugs also, few drugs. But for vaccines, there's been no vaccine that is currently in use which performs differently in different races and different ethnicities. So I think we should not worry too much about that.
Okay, thank you.
Dr. Tripathi, would you like to add something?
Yes, in fact, what is of concern is the mutations that are occurring in the South African strain and the Brazilian strains, because the AstraZeneca has been reported to be protective against the UK strains, but will this protect against the Brazilian and South African strain? That maybe we need some information on this. Maybe if Dr. Kulkarni has some information on this.
So actually for the UK strain, obviously it has shown clinical efficacy. Also for the South African strain, it has shown clinical efficacy. And also the in vitro studies have shown that the sera from the vaccine trials can neutralize both these strains. So as of now, there is no indication that this vaccine won't work against these new strains. Okay, so as of now, there is no matter of concern. Let's see how the situation unfolds, and time will only tell. But as of now, there is no need for concern.
Would a third dose improve the efficacy of the vaccine to close to?
No, actually, you have seen that after the second dose, the titers were so high that in fact they are higher than even the naturally infected people. Yeah? So antibody titers up to 33,000 IgG. So that's a very high level. So I don't think adding a third dose would make any difference, because the immune response might have reached to a saturation point.
Thank you.
To Dr. Prasad and Dr. Tripathi. I saw two hands go up. Let me take the first one first. Shobana, are you around? Shobana, a couple of questions were being rolled out from you. I saw a lot
Yes, I am around.
Shobana?
I am
Yes.
here.
The floor is yours.
Yes. I have a question, and whoever is appropriate can answer the question. As a user, as a prospective user, my worry is, when are the results of the phase 3 clinical trials for Covaxin expected? Does Dr. Kulkarni have any information on this?
Not really, because the trial is still going on. That's all I know. That's the only information that is available in public domain. So let's hope that the results will come soon, because this is an efficacy trial, which means that they need to get the required number of COVID cases in the trials. So it is not really about the time of follow-up, but it's about how soon they can get the cases. So,
but I heard, I'm sorry to interrupt, but I heard that they have the required numbers. I was reading a report in the Hindu which said that they had managed to amass the number of participants required. So
it's not about the number of participants, it's about the number of COVID cases that happen in those participants.
Okay.
So I don't know. They may have recruited everybody, but they need to have maybe 100 or whatever number of cases to happen. Once they reach to that time point, then only they will unblind, the data, and then they will get to see how many cases of those 100. Let's say, I'm just giving an example. I don't know how many cases they require, but let's say if they require 100 cases, and after they unblind, then they will find out that, okay, maybe 80 cases are in the placebo group, 20 in the vaccine group, which means that the vaccine is showing really high efficacy. So that's what it will tell us, right? So we will have to wait till they hit that number of cases, required number of cases.
Understood. Thank you very much. Right. One more question, if I am allowed to ask you, Dr. Kulkarni.
Maybe you can come back, Shobna. There
Sure.
are a few people waiting.
Sure. Sure. Yeah.
Okay, the next question will be from Mrs. Lakshmi Swaminathan, who has been having her hands up. I hope it's not hurting, madam.
Thank you, Jayanti, for giving me the floor. And, I must say it was a very informative discussion, and I thank all the panelists for educating us and to appreciate the problems as well as the cure for this COVID-19. Now, my question is that we've been seeing in the press that in states, for example, like Kerala and Tamil Nadu, which are supposed to be having high rates, the number of responses or people coming up for vaccination is somewhat low, and government was there, central government was therefore trying to enhance the efforts to get more people for vaccination through participation of celebrities from various fields and so on. I mean, in addition to that, I would like to know whether any of the panelists have some suggestions how to mobilize and try and get these people to come and vaccinate, because we all understand that it is necessary and it is important. How do we get the masses to come in? Thank you. Any one of you.
Dr. Prasad Kulkarni could answer the question, please.
Yeah, I think government is trying its best to, instill confidence in the minds of the people, and I think that is actually making a difference. Already 54 lakh healthcare workers have received the vaccine, which is not a bad number at all. And slowly, obviously, this number will go up further. So eventually, their target of, I don't know whether 1 crore people, I think that will be made in coming future. So only thing I think what one can do is just keep on spreading good word about how important it is to take the vaccine, especially on the social media and all that. All these doctors who have taken the vaccine, many of them actually posted their pictures with of taking the vaccine and how they are feeling nice and safe and everything. I think that should spread around. This will really give more and more confidence to others. That's very important, because maybe in a month or so, the vaccination campaign will start for the elderly people and those with comorbidities. So there, these people should be confident to take the vaccine, because so all these efforts from the doctor community will really go a long way in doing that.
Thank you, Dr. Kulkarni. There is a question from Dr. Ramakrishnan Ganapati. Please, over to you. Please unmute yourself, doctor. Dr. Ramakrishnan Ganapati, please.
Yes, very good afternoon to you. Really, it was quite enjoyable to listen to everybody. Maybe I think that I have two questions, short questions, and I hope that maybe Dr. Kulkarni or Dr. Tripathi would be able to answer this. Now, based on the way that some of these vaccines are made today, the mRNA approach is one of the latest technology, and then the other ones like the viral vector is a conventional one, and then you have the others using the whole virus and the protein, etc. So one first question would be, which approach is much easier or much more economical to do in a rough way? I mean, not in a precise way, but at least as an approximation. That would be my first question. My second question would be, I understand that J&J is coming with, that is Janssen is coming with a new, another vaccine introduction. And this vaccine is supposed to be a single dose vaccine. Now, how are they able to do this as a single dose vaccine against the two dose vaccinations? These are the two questions I have.
We could have the question answered by Dr. Shrikant Tripathi. Could you please unmute yourself, Dr. Shrikant?
Yeah, I've unmuted. Can you hear me now?
Yes, doctor, we can.
Okay. As far as the logistics are concerned, this Pfizer vaccine has to be stored at minus 70 degrees. And minus 70 degrees, we really don't have the infrastructure in our vaccination program for storing this vaccine in large numbers all over the country. And if you go to the rural areas, the things are even worse. Some of the rural centers, I'm sure, would have never seen a minus 70 degrees freezer. This may be probably available in some research laboratories, but in the general hospitals or in the PHCs, most of the hospitals just don't have minus 70. So storing Pfizer in our infrastructure will be a very difficult proposition. And so from that angle, Pfizer looks very unlikely in our setting. Comes minus 20 again. We have 4-degree freezers in our PHCs. We have 4 degrees centigrade in our hospitals. At minus 20, okay, there are places where they do store polio viruses at minus 20 or minus 40. Those facilities can be utilized, but when you're talking of huge numbers, then the infrastructure. So 4-degree, those vaccines which can be stored at 4-degree would be easier to use in our setting. And as far as sorry, what was the second question? One is the logistics and the cost factor. You said.
Johnson. Yeah. Second one was about the
single dose. Yeah, single dose, right?
Single dose versus double dose.
I would request Dr. Prasad Kulkarni to answer that. Why AstraZeneca we cannot use the single dose? Because I think this all depends on the response we see with the single dose, and is there an adequate antibody response? And if there is a requirement for a booster to increase neutralizing antibody titer, I'm sure the basic science researchers have worked out this, and that is why they have advised 2 doses. But then can it be modified to make it a single dose? That I think only Dr. Prasad Kulkarni can answer that question.
Yeah, I think they have given their data, and it looks like something like 65% efficacy with single dose of the vaccine and around 85% for the severe disease. So for severe disease, all other vaccines have generally almost 100%, including the Oxford vaccine. So I would say on a little lower side. So my guess is that even they will benefit if they add a booster. Right now, they are projecting it as a single dose vaccine, for which they have got the emergency use authorization from US FDA also. But it is quite possible that if they add another dose, their efficacy will go up much further.
Thank you very much. The only question was that minus 78 storage, we have plenty of that, but transportation is a bit of a problem. Minus 78 storage is all the blood banks are using. My question would be that, what are the cost of production of these different technologies?
It's very hard to say. It's really, we are, because unless we have their internal data, nobody can comment how much is their production cost. So, and moreover, their production is happening in US, and so many other factors are there. So it's really very difficult to comment. The technologies, though may sometimes appear very attractive on paper, can be really complicated in real life to make. So it's really hard to comment on that.
Thank you.
Thank you, Dr. Prasad Kulkarni. We move over to the next person on the queue, Anita Bogle. Are you here? Please unmute yourself, Anita.
Yes. Firstly, thank you to all the speakers for all the information. But this is like a little confusing for a lay person. first of all, we've got to register, and then, in a few months, we will probably be eligible for the vaccine. And then you say there's only 60 to 70% efficacy, and then you've got to have 2 doses which are spaced out. And then they will be effective only for 6 to 8 months. Are we going to be in this continuous, cycle for vaccines? Is that how it looks?
So I think we have to look at all this data that you are seeing with the correct perspective. First of all, the vaccine is preventing 100% the severe disease, which is the biggest plus point of vaccines, right? We are all scared of the disease because it can become severe. If it was just like common cold, we wouldn't have even bothered of taking a vaccine, correct? It is because of the severity and because of the potential for death that's why we are worried about the disease. And that's, if that is getting prevented, one should definitely go for taking a vaccine. So don't worry about 60%, 70%, 80%. What is most important is that it is giving 100% protection from the severe disease. Even 60%, 70% efficacy is not small. Please remember that even for other vaccines, no other vaccine gives 100% protection, right? There is no vaccine in the world which gives 100% protection from an infection. It can vary from 90%, 95% to even 60%. For example, for rotavirus vaccine, it can be 60, 50%, right? But still, it makes sense to give this vaccine to everybody for 2 reasons. One, it gives individual protection to some degree, which is very high degree. And secondly, as more and more people get vaccinated, you block the transmission of the virus. And if you stop that transmission, then naturally it gives rise to herd immunity. And even those who are not vaccinated also get protected. And this is very important. If we want to stop this pandemic, it is important that more and more people get the vaccines so that we can stop this chain of transmission.
No, so do I hear you say that, as many people as possible must take the first round of vaccine, and then we kind of see how it goes before we know when we need the vaccine again?
Yeah, see, again, as far as the duration of protection is concerned, obviously the data is yet to get generated, right?
Yes.
Which is true even for the infection. But with the infection, we have reports which says that the response is there for 9 to 10 months at least. If that is the case with the infection, and same must happen with the vaccine because the titers are on the same lines. But this does not mean that the protection stops at 9 to 10 months. It is just that we are still not getting the data. The data will keep getting generated, and we'll have more and more information about duration and protection. But going by the titers that we are seeing, my guess, my personal opinion, that the protection is going to be very long lasting, very long lasting. You saw the antibody titers like 33,000. These are very high titers. So they are going to take you for a very long time. It's not also just about the antibody titers, but also about the T cell responses, your immune memory. So many a times for many vaccines, even if you lose all your antibodies, still your memory cells are active. The moment you are exposed to the same virus, you get protection, right? So I think the vaccines are going to give fairly long-term protection. And if more and more people can get vaccinated, the virus can go away as soon as possible. See, this is what had happened with the MERS epidemic that came in 2011. Also with the SARS epidemic that came in 2003. Both of them were coronaviruses, remember. So the virus also belongs to the same family. So my guess is that if you achieve a good amount of coverage of the vaccine and already infected people are there, the virus may go away, may simply go away. Let's hope for the best. As of now, yeah.
Thank you so much.
That was an eye-opener, Dr. Prasad. Thank you very much for that. We have the next question from Munuswamy Shekhar. Munuswamy Shekhar, please unmute yourself and you may ask your question.
Thank you, madam. So my only question is that vaccinating, does it ensure you can stop your cab, as ma'am so said, like the COVID appropriate behavior?
No, no, not at all. So even if you take complete vaccination, still you must continue taking all your routine precautions.
So the other one I just put was that the slow response in Tamil Nadu especially is a concern. And as ma'am already told that, I don't know what could be the reason, because here the 3 C's also, which other senior ma'am said, are not being followed. We are able to see the crowding. We are able to see the people close contact. We are also theaters opened and closed. This thing also closed environment. People are there. So how do you see this is going to affect our all our business?
Really hard to comment why the vaccine uptake is little lower. I'm sure the better sense will prevail and more people will come up and come forward. As far as people not taking precaution, I think it's not only in Chennai, it's happening everywhere. Everywhere people have stopped, many people, not everybody, but many people have stopped taking precautions, which is not good at all actually. You never know when the virus will come back again. This is what happened in Europe. They became complacent and then it came back with a vengeance. So let's not lower our guard. We should continue taking precautions.
Yes, doctor, we understand that, especially the educated ones understand.
Yeah.
But I think, is it not the duty of the government to see it still more stronger? They come down and then say, no, you have got to be still going on. Because again, they see from the government side also, it is slightly lax and all that. So automatically people also now relaxing.
I think Dr. Kulkarni may not be able to answer that. So go on, Jane.
But we have to be very careful.
Yeah, absolutely. Go on, Jane.
Thank you.
That was a good comeback on that. And we have the next question from Mr. P.V. Srinath, who had his hands up. P.V. Srinath, could you unmute yourself, please?
Yeah. Hi. Yeah. Excellent presentations by all the speakers. My question was essentially that all the vaccinations which are currently being developed are injection based. There was a talk sometime back. I was reading about some nasal sprays, but nothing has come out so far in terms of actual vaccine. Is that because of some concerns about that, or is it just the time taken for that is likely to be far more than what is taken for other vaccines? And are there any pros and cons of that?
So, as far as the nasal vaccine, in fact, just Bharat Biotech got their permission for clinical trial for this thing, for their nasal vaccine. So that development, Bharat Biotech development is going to continue. Obviously, it will take time to come to the market actually, but that is going on. Then there is another second intranasal vaccine with which Serum Institute is also working in collaboration with a company called Podagenics in USA. This vaccine is already in a phase 1-2 trial in the UK. And after this trial is finished, then obviously we'll do the phase 2 and phase 3 trials. So this vaccine again will take some time. Most probably by mid next year, this vaccine will come in the market. So the development is on. There is nothing that has stopped.
Thank you.
That was a good reply to the question, because none of us, it's only a kind of speculation that is happening now. Okay, now let's go for the last question to Radha Vijayaraghavan. You could unmute yourself and ask your question.
Good evening, sir. My question is to Mr. Prasad Kulkarni. I've read in the fact sheets of some of these, the 2 vaccines in India, that people with allergies to medicines, etc., that's a contraindication to take the vaccine. If so, what are the things that we should be wary about? What are the things we should check with the physician, and which kind of allergies should we check with the physician before taking the vaccine?
Yeah, so if you have read the Covishield fact sheet, it clearly says that if you are hypersensitive or allergic to any component of the vaccine, then you should not take the vaccine, right? Anything, the list of the constituents of the vaccine is also given in the fact sheet. Only those allergies are concerned. If you are allergic to, let's say, other medicines like penicillin, or if you are allergic to some food, like eggs or fish, that's not a problem.
Yeah.
That's not a problem. You can take the
Where are the components mentioned in the fact sheet? I'm sorry, I couldn't find it there in the fact sheet which was presented on the TV.
They must be there, or they are also there in the package insert, which is there with the doctors.
Oh, the doctors would be knowing.
Yeah.
Okay, thank you.
Very much for that, Radha. I think the question on most of our minds now is how to get the masses to understand the importance of the vaccine. And perhaps one of the ways to do it would be to request WITT and MCCI to organize more seminars of this kind so that we could actually accept all the questions of all the hands that have been raised up. Thank you very much for your participation.
Jayanti, can I have one comment, please?
Yes.
Jayanti, just one comment and one lighter vein of a question. The comment is, India is now, by and large, a major exporter and also an importer. So with the result, some of the intellectual property issues that we were mentioning will have to be looked at from both sides. Because at the moment, maybe a few years back or 50 years back, we would have imported most of the things. But today, we are also exporting quite a bit of the, at least in the vaccine, the so-called vaccine diplomacy, if somebody has used, definitely we are an exporter too. So therefore, I think we are in a very enviable position compared to many other countries. And so I think we should be very proud of that situation. This particular feature may be of interest to the Madras Chamber of Commerce and Industry, because there we have both the industry people and the commerce people. They may have a quick look at this and to see what role they can play in the further production of the vaccine or in the further export of this vaccine. So this may be one important thing that we have learned. I am sure Saraswati would have noted this already. On a lighter vein, we had a question sometime whether it is 1 dose, 2 dose, or possibly even a higher dose. I was only wondering, Dr. Guleria, how many doses he must have taken, because any day, every day we were seeing he was having 1 prick or say even 2 pricks on the same day. I hope that will not do him harm. Thank you very much. In the TV only, not real. Okay.
Thank you very much to all for participation in this seminar. Perhaps we should actually hope and wish that WIT and MCCI should come together on a repeated basis so that we could actually take more and more questions. Actually, we are preparing a handout on the subject. And if you're interested, you could please mail WITT. The email address is available on the flyer. Please mail us and you would get a synopsis. You could also go through the YouTube where you could actually replay these speeches. And if you come across more questions, you could definitely email the speakers. And indeed, it was a memorable seminar for each one of us gathered here. And now may I request Mrs. K. Saraswati, the Secretary General, to deliver the vote of thanks.
Thank you, Jayanti. You have done most of my part. So I have very little to do. I'll do my best. I have made note of Dr. Swaminathan's suggestion. Of course, it is indeed a pleasure for the Madras Chamber to join hands with WIT and have this kind of intellectual programs. And maybe in the healthcare, this is the second program, but we have done much more, as you know, on ocean economy, things like that. And we have always felt it's a very, very useful and very intellectual discussions, and at the same time, very grounded. We could see that once again here. So Dr. Swaminathan, definitely I've made note of all your points. With your blessings, we'll definitely take this forward with your support as well. Let me just very quickly sum up and probably give a very quick vote of thanks since in the interest of time. I should first thank Dr. Bhargava. In fact, I recall with pleasure, we had her in 2019 when we had the medical leadership program. Probably none of us thought about this COVID and this virtual thing. So we thought we are going to again meet again in person. But we are happy and thankful to God that we are meeting virtually. Technology has facilitated that. God has facilitated that. Thank you so very much, ma'am. It was a very, very wisdom-filled address, I would say, as we found always. And as you mentioned about the CAP, the COVID appropriate behavior, someone also mentioned, I think that has to continue. Of course, Serum Institute cannot take the responsibility of enforcing it through the government. But yes, every one of us are responsible. And also, you mentioned very clearly the 3 C's to avoid: close contacts, crowded places, close premises. We will have to promote and probably enlighten people more on that. And you also talked about the different approaches by different countries. Maybe we'll have to wait and watch. India, probably we are prohibited. We will take only one, not the cocktail. Only one vaccine, probably both the doses. But as you said, there are other countries who are also going through the cocktail mode. And also you made it very open, saying the duration of the effectiveness of the thing, it's still anybody's guess. We are still not clear about that, but that has to be that way for some more time, because the whole thing is so nascent, so new. So maybe as we go along, we will learn. Thank you so very much, Dr. Bhargava. It's indeed a pleasure, and we look forward more and more interaction. Now the healthcare committee is also becoming very active in the chamber. Thank you, madam. And I should thank Dr. Manimanan, who is not here, but he set the right tone, talking about the India's resilience, because every country, when the COVID broke, and even people in India, we were all worried, given the population, given our kind of hygiene factors, would we be able to manage this or win this battle? But whatever we have to be the reasons, we have really done very well, and we can be proud about that. The resilience is good, but it should give any complacency. But at least we are happy that we could prove to the world that we could win this battle also. He made the tone right. And he also said giving awareness and building confidence is more critical now, as every one of you and Ms. Jayanti mentioned it again and again. Probably that is very critical now, because it's very new. As he said, for children, vaccination, we are all used. For adult vaccination, when it comes to that, we start thinking like adults. We say, oh, should I go beyond? So many questions. And there is this WhatsApp University and Facebook University to confuse us further. So probably this is time for the chamber, for every organization, which and all of us will join together and see how to create the awareness. So I should thank Dr. Manivannan and Mr. Tripathi. As I said, when so much information is floating in the WhatsApp and things like that, was a very, very clear approach, very clear kind of a presentation on the evolution of vaccines, comparison of different vaccines, the quality, safety, immunogenicity of and efficiency of the vaccines, and the fact sheet about the vaccines. That's really great. Thank you so much for joining us. And what should I say about Dr. Prasad Kulkarni? The Serum Institute is most sought after now, and even here in the thing, so many questions posed to you, because everybody, in fact, I contained myself not asking any questions because it was going beyond the time limit. Maybe we will have to take it up separately. But such a complex subject, such a complex virus, you made a very simple, very sort of easy to understand kind of a presentation. And probably the good news you said is for severe cases, it's 100% effective. We all are looking for that. And for other cases also, it is around 70%. As you said, any percentage is good now because we started with 0. So we are very happy that Serum Institute is doing that. We are extremely thankful to the Institute. We are thankful to you for being available here and having the patience to take so many questions. There are some more waiting, which we will have to probably forward to you later. And Mr. Bansal, as Dr. Swaminathan said, we owe him a special thanks because we got him at a very short notice. And he started with a disclaimer. I don't think there is any need for this disclaimer. He made it very simple, very clear about the thing. And so the serological aspects, especially since he is working with the immunocompromised group, how important it is and what would be the correct thing and all. So he closed with the Tarizamin per slide. I think it's good. Probably we are also trying to count the stars in the sky, and probably that will happen once we have the vaccination process in full swing. And what is more important after all the thing is our intellectual property. We have so many intellectuals here. Who are we going to protect? So Dr. Kaddam, Dr. K.S. Kadam made it very clear when the developed countries and the developing countries, the concern for us, when with less population, they try to pre-book. There is something that we will have to do. And here where I think the real Atmanirbhar comes. the local manufacturing. And you rightly said about the Indian Patent Act, which is very strong. And probably the section 47, if I am right, what you mentioned, the special provisions. And you are ending your speech with the confident note that India is prepared, even if others are not going to help us with that, is very, very encouraging. I think that's a very positive note to get it from the speaker of the day. Thank you so much, Dr. Karna. And the other people, I shouldn't say that. I should start with Dr. Swaminathan, but pronounce to you, sir. You know, the kind of help you have been doing, the teamwork, the kind of information you are sharing, we are always amazed at your knowledge level and your patience level. Thank you. As Ms. Jayanti mentioned, you moderated the session so very well in a very democratic way, giving opportunity for every speaker to share his or her thoughts. And thank you so very much. We would be very, very happy to continue the relationship with the quick-witted wit. And I should thank Dr. Jayanti, who moderated it so well. In fact, I was a little worried about the time management, though we have exceeded, it is for a good cause. And you had really managed the sessions very well, ma'am. Thanks a lot to you. Probably, as I said, with wit, we are going to continue our relationship. And I would thank all the participants who are here. Many people who had asked questions. Our apologies if we had not answered any of the questions, or if you couldn't take up any of the questions, please feel free to share with wit or the chamber. We would definitely do as this, as Ms. Jayanti said, probably we will come with an FAQ kind of a book, and that would be helpful, and that would also create awareness. It could be a twin-edged kind of a thing. So our apologies to those whose questions we couldn't take up, but probably more and more will be coming, and probably this is just the first of its kind. We are going to have more such awareness campaigns. And also I thank my colleagues who are working from behind the screens for enabling us to be in the screen. Thank you so much, Swarna, Madhu, Jaya, Basmati, all of you. And also, if someone from press have joined, I'm not very sure, but thanks everyone. Have a safe day. Have a vaccinated day as early as possible, probably, and have a good weekend. Thank you so much.
Thank you so much.
Thank you.
Thank you.
Thank you very much. Thank you for everybody. Thank you, sir. Thank you, ma'am.
Thank you, sir. Thank you.
Thank you so much, Mrs. Saraswati, for your big rollout of meticulous thank yous to each and everyone. That was really wonderfully done. And perhaps one of the best things we could do is, if I could request the president or the chairman of WIIT and Dr. K.V. Swaminathan and the president of the MCCI to perhaps put this YouTube and make it go viral, because I've been seeing these comments by many people who said this should be uploaded on the YouTube and every citizen of India should see it. So perhaps this will factor in many a mind. It did in mine, right? And I'm sure.
Sure. And sorry, my apologies. I didn't thank enough Mr. Jai Shankar, who had been the brain behind this program right from day one. My apologies, Mr. Jai Shankar. She called you Shankar. We used to call you as PVJ. So thanks, PVJ, for all the efforts. And what you said will be rightly noted. We have recorded it. Normally, unless the speakers have any objection, we will straight away take it to the YouTube. Today, it would be available.
Yes.
In fact, I have shared it with my general committee already, the YouTube.
Thank you. Thank you.
I would dare suggest that this would be a need of the hour. Need of the hour thing for us to do to bring about this awareness with the masses. And thank you to all of you for joining us in this eye-opener session. And may I now request all of you to rise for the national anthem. Swarna, over to you, please.
Just stand up. You look national anthem.
Can you play, Swarna? Or should we sing? Should we have our audio and sing? Or you
अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंधु गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जन गण मन
Second time. Thank you very much.
Thank you, one and all.
Thank you all.
Thank you, Jayantiki. Thanks.
Thanks everyone, ma'am. Thank you. It's a very great event, fantastic event.
Thanks a lot.
Dr. Swaminathan, sir, thank you. Many of our former professors joined.
Thank you, sir. Thank you, Dr. Shekhar. Thank you. Thank you.
Thanks to
Thank you, Professor. Thank you, sir. Thank you, madam.
Saraswati. Thank you.